During this session on the special senses, we're going to think about the ears, but in this case, we're going to be thinking about hearing rather than balance. So by the end of the session, we should be able to think about the basic structure and function of the ears, relating that to hearing itself. And looking at the major areas of the ear that are involved in hearing, there are three parts. We've got the external outer ear. That is only involved with hearing. We've got the middle ear, which is where we have the tympanic cavity, and it's the tympanic membrane or the eardrum that basically connects the outer ear into the middle ear. And this middle ear is air-filled, and it has three little bones, the stapes, one of which is the stapes, the smallest bone in the body. But these three little bones, the auditory ossicles, they form like a lever system to help to amplify the sound. So when sound comes in through the external acoustic meatus, which is also known as the ear canal, the tympanic membrane starts vibrating. That causes the three little bones to start vibrating, and that helps to amplify the sound before it then goes into this area, which is the oval window, which connects onto the internal ear. And the internal ear is involved in both hearing and equilibrium, so it has both sets of apparatus there. And they are separate, independent receptors for hearing and balance, and they are the cochlea when we're thinking about sound and hearing, and the vestibular organs when we're thinking about balance and equilibrium. And of course, we also have the auditory tube that connects down to the pharyngeal region. And this is where you can get you have equalization of air pressure, and that between the middle ear here and the external environment. So you get that equalization of air pressure by moving the jaw and swallowing, because it lets air into that auditory tube. Now, when we're thinking about hearing, we do have to think about how do we actually localize sound? How do we know where it's coming from? Well, if you're thinking about sound coming from your right-hand side, for example, you know that the sound is going to arrive at your ears at different times. So if it's coming from your right-hand side, it's going to hit your right ear first. So there's a time differential between the sound hitting the right ear and hitting the left ear. The sound is also less intense as it reaches the further ear, because the head is acting as a sound barrier. And so the auditory cortex is able to integrate these cues and pinpoint the location of the sound source by the differencing in the timing of neuronal firing coming from each ear. So what is the relationship between the physical stimuli and sensory perceptions? Well, sound from whatever it might be hitting the table, or from your voice, or from cars, or from aeroplanes, leads to pressure fluctuations. Those pressure fluctuations are sound waves, and that is then picked up by the middle and inner ear, and it's transformed into mechanical and then onto electrical signals before it goes to the brain via action potentials for processing, where we start to interpret and we start to recognize and we start to give meaning to it. There are a few little interesting facts for you. So one of the middle ear bones is the smallest in the body. That's the stapes, and that is the smallest middle ear bone, the auditory ossicle. The eardrums moves less than the diameter of a hydrogen atom for minimum audible sounds. That's quite a small amount. The inner ear reaches full size when a fetus is 20 weeks old, and hearing loss is the #1 disability. So about 75% of people have some form of hearing loss during their lifespan. It's also important to remember, and I think you'll all know this anyway, that sound can travel through media other than just the air, for example, water. However, due to the greater pressures required to cause fluid movements, sound transfer is less efficient in those mediums, and there's greater resistance to change. So what's actually happening? Well, the cochlea is acting as a frequency analyzer, and it operates on the incoming sound's properties. So we have intensity. So intensity is measured in decibels, and it's perceptual. If you're thinking about what it is, it's all about loudness. And this decibel scale is somewhere between is up to about 120 decibels. And sounds greater than 110 can actually permanently damage the cochlea. We then have frequency. And the frequency is just the number of cycles per second, or the number of waves per second. And that gives us our pitch or our tone. So our E or our O. And of course, decibels is loudness. Loud, quiet. Sorry, just being a bit silly, but that's just me. Anyway, so the frequency of the human ear is somewhere between 20 and 20,000 hertz. So that's the kind of range that we have, though that does change as we get older. So the greater the pitch, the larger the frequency of vibrations. And then you have complexity, which is all about timbre and quality and different sounds coming together. So the cochlea contains receptors that help to convert sound waves into nerve impulses, and the vestibular apparatus, as we know, is necessary for equilibrium and balance. So we have these complexities of sound. We have pure tones where if you're looking at this one, we have a nice, simple, periodical, regular sound of a single frequency. We then have music tones, we'll know about those, fundamental frequency with many harmonics, and then kind of noise, random tones, if you like, and sounds. But remember, our ears are processing information all the time. The rustling of the leaves, the birds tweeting away, cars in the background, someone talking to you, all that information is hitting your ears, and we have to be able to interpret appropriately and get rid of and filter out the information that we really don't want to be listening to. So when we're thinking about the structure and function of the ear, we've got the external outer ear, which is all involved with hearing. This is also known as the auricle or the ear, and it funnels the sound waves in through the external acoustic meatus. Remember, that's also called your ear canal, moving towards the tympanic membrane, which is the boundary between the external ear and the middle ear, where your auditory ossicles are. Now, the external ear also has skin bearing hairs and sebaceous glands that secrete wax. And those glands are basically modified sweat glands, and they help to provide a sticky trap for foreign bodies and repel insects. This wax is naturally removed without being noticed, for example, when you do jaw movements via eating and talking, and it falls out of the external acoustic meatus. In some people, it does obviously compact and build up. And actually, just so that you know, if you're sharing a bed with someone, you're sharing a pillow or you're very close, a lot of this wax does actually fall out at night onto your pillow, something very nice to know about. You've then got that tympanic membrane, that's also called your eardrum, and that's, as I've said, the boundary between the external and the middle ears. And the middle ear is an air-filled cavity. And the sound waves that are coming in through the external acoustic meatus make the tympanic membrane vibrate. And then those tiny little auditory ossicles, those tiny little bones of the middle ear, vibrate at the same frequency. And that sound energy is amplified and transferred to the oval window, which is just there in the image, and that vibrates at the same frequency. And that then goes on to cause the cochlear fluid in the inner ear to get set into a wave motion at that same frequency. So really, if we're thinking about it, it's quite simple. We've got sound waves coming in, causes the tympanic membrane to start vibrating, causes the auditory ossicles to start vibrating, causes the oval window to start vibrating, which causes the fluid within the cochlea to start forming waves. And then that causes hairs to start moving, and we start to get action potentials. So if we're thinking about all of this, the sound energy is amplified. And it's amplified in a couple of places, from the outer to the middle ear by the lever system of the three auditory ossicle bones, and by the size difference. The tympanic membrane is about 20 times larger than the oval window, therefore the pressure is about 20 times greater. But we've also got to remember that for the tympanic membrane to vibrate freely, the resting air pressure on both sides of that membrane must be the same, and I alluded to that a little bit earlier. Both sides are connected to the atmosphere, the outside via the ear canal, and the inside via the auditory tube, which is connecting the middle ear to the pharynx. And this tube is usually closed, but can be opened by yawning, chewing, and swallowing, as we've talked about, and it helps to equilibrate the pressures, for example, when you're descending in a plane or going diving. And during rapid pressure changes in that eardrum, that causes the eardrum to bulge painfully, in particular as the outside pressure changes, but the inside doesn't, and that's what's causing the pain. And infections originating in the throat sometimes spread to the middle ear. You get fluid accumulation, and that becomes painful, but also it starts to interfere with sound conduction. So the cochlea itself is a coil tube with fluid-filled compartments that contains the organ of Corti. And that's the sense organ for hearing, with associated hairs that can move as the waves move. And so what's really happening is you're getting the sound coming in through the external acoustic meatus, causing the tympanic membrane to vibrate, the auditory ossicles to vibrate, the oval window to vibrate, and that causes the perilymph to vibrate. Now, the perilymph itself, the size of the waves depends on how loud it is. So the louder it is, the bigger the waves. The quieter it is, the smaller the waves. And so when this crosses the basilar membrane and causes the organ of Corti hair cells to move, if it's a bigger wave, it causes them to move more. If it's a smaller wave, they move less, and you get more or less action potentials because of that. And at the same time, you have different regions of the cochlea that respond to different frequencies. So the low frequency sounds stimulate the hair at the far end, whereas the higher frequency sounds are being stimulated near the base, near the beginning of the cochlea. Sorry about the silly voice, by the way. So really, what's actually happening is the cochlear fluid in the inner ear sets into wave motion at the same frequency as that of the oval window and the auditory ossicles and the tympanic membrane. Fluid movement can go two ways. It can follow the perilymph all the way around. So here it is. Here's our perilymph starting at the oval window, and it meanders its way around and heads off to the round window, which then bulges out and the sound energy is dissipated. It can also cross the basilar membrane and start to stimulate the organ of corti hair cells, and that by triggering those, which are obviously going to be mechanoreceptors, so they're hairs, they're moving due to the pressure changes with the waves, and that will eventually lead to action potentials, traveling down the vestibulocochlear nerve, and then that's obviously going to your auditory cortex where it can get processed. And if you stimulate these ones at the far end of the cochlea, you have that deeper sound. The nearer ones, you have that higher pitched sound. And of course, you've got all the ones in between. And then, of course, depending on how much those hairs are moved, depends on the loudness, so the number of action potentials coming through. And so really what's happening, different sound frequencies cross the basilar membrane at different locations. And I've already talked about this. So here's our basilar membrane, and here's our cochlea. And so you can see the high frequency sounds, as I've already said, they displace the basilar membrane near the base. The medium frequency sounds displace the basilar membrane near the middle, and the low frequency sounds displace the basilar membrane near the apex. And here it is, just to show you. So at this region near the base, the basilar membrane is narrow and it's got a stiff end near the oval window, and so that's where you have the higher frequency. And then if you make your way all the way down to the apex, it's wider and flexible end of the basilar membrane. So that's for those deeper sounds. And of course, excessive exposure to loud sounds can lead to hairs wearing out, and that can lead to partial deafness. But also, as you'll have found out when you basically tested yourself, you might have not heard the sounds at 20,000 hertz. In fact, you almost certainly wouldn't, because you lose those hairs seem to die off a little bit quicker. So as you get older, your ability to hear these higher frequency sounds starts to dissipate. So what is the auditory pathway to the brain? Well, the organ of corti hair cells form the vestibulocochlear nerve. The auditory fibers cross, and both hemispheres receive information from both ears. And remember, that helps us to work out where a sound is actually coming from. That sound crosses over to the thalamus, then moves to the thalamus, the primary auditory cortex, the auditory association area, where we get an understanding of what's actually being heard. It can also go to the inferior colliculus, so the brains within the brainstem. Remembering back what's happened with the superior colliculus. So the inferior colliculus is the auditory reflex center, where if you turn your head away in response to a loud noise, remember the superior colliculus is your visual reflex center. So that's where a bright light shines on you. You turn your eyes and you turn your head away. So you've got those two reflex centers there. So in a little bit more detail, if you just want to have a quick look at it. So you've got the cranial nerve 8 down to the cochlear nuclei. So if we're just following the pathways here, remember it's coming from both ears. So you've got your cochlear nuclei, then you've got your inferior colliculus, so that's your reflex center for those loud sounds, remember. We're then going to the thalamus, the medial geniculate nucleus of the thalamus, and then on to the primary auditory cortex and the association area, where you get into interpretation of that information. So a couple of review questions for you to think about. So press pause, and then we will go through those questions. Okay, when you swallow to pop your ears, you are allowing air to escape via the. Only one of them is correct. Which one is correct? Number 5, hopefully you said auditory tube. And which of the following are correct? The tympanic membrane receives sound waves from the auditory ossicles of the outer ear. That is incorrect. It's the other way round. The amplitude of a sound wave decreases as it travels along the basilar membrane. That's incorrect. The amplitude does not change. High-frequency sounds cause maximum movement of the basilar membrane near the base of the cochlea. That is correct. Frequency range of human hearing is about 20 to 20,000 hertz. Well, hopefully you got that. That's correct. And the higher the frequency, the louder the sound. That is incorrect. It indicates pitch, not loudness. Remember, it's all about oh. You can always tell I was clearly a great singer in my youth.
The sense of hearing is accomplished by a process known as auditory transduction. The ear converts sound waves in the air into electrical impulses, which can be interpreted by the brain. As sound enters the ear, it passes through the external auditory canal, where it meets the tympanic membrane. The tympanic membrane then vibrates in response to the sound. Sounds of a lower pitch, or frequency, produce a slower rate of vibration, and sounds of lower volume, or amplitude, produce a less dramatic vibration. Higher frequency sounds produce faster vibrations. The tympanic membrane is cone-shaped and articulates with a chain of 3 bones called the auditory ossicles. They consist of the malleus, the incus, and the stapes. The movements of the tympanic membrane vibrate the ossicles, passing on the information of frequency and amplitude. The 3 bones pivot together on an axis shown here in red. The pivotal axis is due to a series of ligaments which hold the bones in place within the middle ear cavity. The anterior malleal ligament and the posterior incutal ligament are of particular importance for the pivotal axis. 2 structures which normally obscure this view of the middle ear have been removed. They are the chordae tympani nerve and the tendon of the tensor tympani muscle. Through the ossicles, the vibrations of the tympanic membrane are transferred to the footplate of the stapes. The stapes moves with a piston-like action, which sends vibrations into a structure called the bony labyrinth. The labyrinth is filled with a fluid called perilymph. If it were a completely closed and inflexible system, the movement of the stapes would be unable to displace the perilymph, and therefore unable to send vibrations into the bony structure. Due to the flexibility of a membrane called the round window, the stapes movement can displace the perilymph, allowing vibrations to enter the labyrinth. The corridor leading to the round window is found within the spiral portion of the bony labyrinth known as the cochlea. Vibrations produced by the stapes are drawn into the spiral system and return to meet the round window. The portion of the spiral passage in which vibrations ascend to the apex of the cochlea is called the scala vestibuli. The descending portion of the passage is called the scala tympani. A third structure, called the cochlear duct, is situated between the scala vestibuli and the scala tympani. The cochlear duct is filled with a fluid called endolymph, and when viewed in cross-section, the membranes separating the 2 fluid-filled systems are visible. They are Reissner's membrane and the basilar membrane. The membranes are flexible and move in response to the vibrations traveling up the scala vestibuli. The movements of the membranes then send vibrations back down to the scala tympani. A specialized structure called the organ of Corti is situated on the basilar membrane. As the basilar membrane vibrates, the organ of Corti is stimulated, which sends nerve impulses to the brain via the cochlear nerve. The actual nerve impulses are generated by specialized cells within the organ of Corti called hair cells. The hair cells are closely covered by a structure called the tectorial membrane. As the basilar membrane vibrates, the tiny clusters of hairs are bent against the tectorial membrane, triggering the hair cells to fire. The entire basilar membrane does not vibrate simultaneously. Instead, specific areas along the basilar membrane move variably in response to different frequencies of sound. Lower frequencies vibrate the basilar membrane closer to the apex of the cochlea, whereas higher frequencies produce vibrations closer to the base. This arrangement is known as tonotopic organization. Together, this sequence of events is responsible for our acoustic perception of the world around us.